Hello, and welcome to Chapter 3, Tools of the Laboratory. In this chapter, we're going to discuss things that you're going to be using in the laboratory and some of the procedures that you're going to be using. And so for this chapter, I chose a picture of what's known as microbial art. And this is simply where we use microbes that produce different colors in different medias to actually draw art. And there's a whole bunch of these available that you can see on the internet. But how would we actually draw these? Well, each one of these different colors are different microorganisms, and we place them in specific areas in order to make the art. So we're going to talk about the tools of the laboratory in this chapter. First of all, I want to introduce you to the 5 I's. These represent 5 basic techniques to manipulate, grow, examine, and characterize microorganisms in the laboratory. The first I is inoculation. This is the introduction of microbes into a growth media. Incubation is where you allow time for these organisms to grow, and we can grow them at various temperatures. Isolation is where we're going to actually isolate a single entity or a single organism from a colony. Inspection, we're going to look at the different characteristics of the organism, and those should all lead then to identification of the organism. So, first of all, you have to collect your specimen. And the specimen can actually be collected by several means. One, you can take it directly from a patient, such as in a wound or lesion. You can use bodily fluids, such as blood or urine. Or in the laboratory, of course, we will be using specimen collections that we give you from growth medias. Then once you have that specimen, you will actually inoculate it. Several different ways that we can inoculate. We can inoculate medias, such as a petri dish that has agar in it. We can inoculate liquid medias of some sort. Or we could even, in the cases especially viruses, inoculate tissues like the bird embryo. All of these samples then will be grown in the laboratory. And this is through the process of incubation. In our laboratory, we have incubators that are set at 37 degrees Celsius. What is 37 degrees Celsius? It is 98.6 degrees Fahrenheit. So 37 degrees is body temperature. If we allowed microbes to simply grow at room temperature, that would be approximately 25 degrees Celsius. We also can set the incubators for higher temperatures, such as 55 degrees Celsius. And this would be for growing organisms that like it hotter than body temperature. Once we have grown the organisms, we then will isolate a single organism, either through using a single culture from a petri dish and subculturing it into another type of media, or taking it and then again isolating it on another petri dish. We then can take a single colony from that petri dish and then I inspect the organisms under the microscope, look for different things macroscopically, such as the color, texture, or size of the growth plate itself or the organism itself. We can then make slides to look at the morphology and characteristic shapes of the organisms themselves. This is one of the things that you will do when you do your unknowns. And then finally, we can go through a series of biochemical tests, immunological tests, or even DNA analysis in order to identify the organism. So, inoculation, producing a culture. A culture is literally the cultivation of microorganisms. Any growth that appears in or on the media after incubation is considered a culture. The medium or media is a nutrient-containing environment in which microbes can multiply. This can include artificial medias or living tissues. Inoculation is again the introduction of the microbes into the media for culturing. We first want our medias to be sterile. That means we want them to be free of all microbes, and the microbes we introduce through inoculation will hopefully be the only ones that will be growing. So sterilization is a requirement for any instrument used for sampling and inoculation. Once we've inoculated the media, we will let it grow. And the usual temperatures range anywhere from about 20 to 45 degrees Celsius. Again, our incubators are set at 37 degrees Celsius. The incubators can also control atmospheric gases, such as how much oxygen is in the incubator or carbon dioxide. Microbial growth in liquid medium tends to actually look either cloudy, or there'll be some sort of sediment after growth, or a scum layer, or we may see a change of color in the media from what it was to begin with to what it was after growth occurs. Microbial growth on solid media, we will see colonies, and these are visible masses of piled up cells. So, for example, Listeria monocytogenes, optimum temperature growth is somewhere between about 25 to 30 degrees Celsius, and it takes anywhere from about 1 to 2 days to actually see growth in the media. Whereas Mycobacterium tuberculosis, the optimum growth temperature is at body temperature at 37 degrees, and it can take up to 28 days for Mycobacterium tuberculosis to actually grow. Microbes have to be provided with all of the required nutrients in an artificial media in order for them to grow. Some microbes require only a very few simple compounds for growth, while others actually require very complex organic and inorganic nutrients to be in the media. Certain microbes cannot be grown on artificial laboratory media and require either cell cultures or host animals, and this is certainly true of the viruses, but even some bacteria will not grow on artificial media. In fact, of the 500 to 600 different forms of bacteria you have in your mouth right now, we have only cultured on artificial media a very small sample of those. Sterile technique is absolutely necessary when inoculating media. You do not want to introduce other organisms into the media. The media and the inoculating tools must both be sterilized. Measures must be taken to prevent the introduction of non-sterile materials such as room air or fingers or other things into the media. And hopefully we will be in class long enough for me to demonstrate some of these, but you will also learn these in the laboratory. Medias are classified by three different categories: their physical state, the chemical composition of the media, and the functional type, or what is the purpose of the media. In the physical state, we see that medias can be completely liquid at room temperature, semi-solid, solid and can be converted to liquid with some sort of process, or solid medias that cannot be liquefied under any circumstances. Chemical composition would include those that are chemically defined, and these are always synthetic medias. This means we know exactly how much of each ingredient there is in that media and how much is exactly available to the microbe for their growth, versus complex medias which are not chemically defined. In other words, this would be something like a meat broth. How much of the meat was actually digested by the digestive enzymes, how much is exactly available to the microbes, we can't really say for sure. And many of our medias are actually complex that we use in the laboratory. Functional types. General purpose medias, this can be used to grow most any type of microbe. Enriched medias have specific things added for a particular microbe's use. Selective medias will allow certain microbes to grow while excluding others. Differential medias will actually show differences in the growth of two different microbes, either by changing the media itself or the colonies, or the growth pattern looks different from the two organisms. We can use anaerobic medias, which take out the oxygen within the media for those anaerobic bacteria. We can see specimen transport medias, and oftentimes these will be medias that are chemically defined that will be able to keep the microbe alive while we're transporting it through mail or whatever. And then there are assay type medias. And then finally, enumeration medias, where we can actually use these medias to help us count or estimate the number of organisms that are present at any given time. Some medias can serve more than one function. For example, a media such as the brain heart infusion agar or broth that we use is both general purpose and enriched. It is a general purpose media, has pretty much anything that a microbe might want to use to grow, but we've also put in specific parts of brain and heart from usually cows. The mannitol salt agar is both a selective and differential media, and blood agar is both enriched and differential, and I'll give you some examples of these. So, liquid medias. These are water-based solutions that do not solidify at temperatures above freezing and flow freely. Most of the liquid medias that we use in the laboratory will be in test tubes with push caps on them. The push caps will allow oxygen to enter into the interior of the tube. Be aware that these push caps do not seal the tubes, so do not pour the E. coli tube upside down because you're going to have leakage. Liquid medias include broths, milks, and infusion medias. Semi-solid medias exhibit kind of a clot-like consistency at room temperature. Think kind of semi-solid jello. Okay. These contain enough gelatin or agar to thicken it, but not actually produce a firm surface. And most of the time we use semi-solid medias to determine motility of a bacteria. Can they actually move through the media as they are growing and multiplying? We can also use semi-solid medias to localize a reaction to a specific site. Solid medias provide a firm surface upon which cells can form discrete colonies, and this is our Petri dishes. They are usually used to isolate bacteria or fungi, and these are just some examples. So our liquid medias, and while this particular liquid media has a cap that screws on, ours will have push caps. These will all be liquid at room temperature. Semi-solid medias, can't really tell from the pictures, but you'll notice that actually we have done a stab line straight down and straight out with what we call the inoculating needle. And we can see that basically where we put the bacteria is where they're growing. They didn't move throughout the media. Whereas this one, number 3, we did the same thing. We did a stab line straight down, straight out. And as the bacteria grew, you'll notice that we don't really have that line anymore. The reason for that is the bacteria have migrated throughout this semi-solid media. And then the solid, reversible to liquid, it's a solid in the tube at a specific temperature, but then we can change the temperature either by cooling or heating and actually change that to a liquid state. Now, what exactly is agar? Well, agar is a complex polysaccharide sugar isolated from the red algae called Gelidium, and then since we call it an agar. It is very similar to the proteins found in gelatin. It is solid at room temperature, but it will liquefy at 100 degrees Celsius. And once liquefied, it does not begin to solidify again until it reaches about 42 degrees Celsius. All of our Petri dishes All of that media goes into our autoclave, and we autoclave it at 121 degrees Celsius, which means it comes out liquefied. We then pour that hot liquid into the plates and allow them to cool to room temperature. As we reach about 42 degrees Celsius, the media begins to actually solidify, and by the time it reaches room temperature at 25 degrees Celsius, it will be solid. Any media that contains anywhere from 1 to 5% of this agar usually has the word agar in its name. So we can have brain heart infusion broth, which would be the liquid form, same ingredients except it simply doesn't have agar in it, or we can have brain heart infusion agar, where we've added the agar so it will solidify at room temperature. Now again, defined or synthetic medias, these are medias whose exact chemical compositions are known. I know exactly how much of an ingredient is in there, and I know exactly how much is bioavailable to the microbe. Many contain pure organic or inorganic compounds that do not vary from one source to another. They have a molecular content that's defined by the means of an exact formula. Some of the medias that we do use in the laboratory will be defined medias. However, they're primarily useful in research facilities. More often than not, we're going to use complex medias. And these contain at least one component that is really not chemically definable. Our brain heart infusion or BHI, broths or agars. These are complex medias because some of the proteins within that media are broken down by enzymes, and exactly how much is broken down to be available to the microbe, we're not completely sure. So typically complex medias will contain extracts of animals, plants, or yeasts. They may contain ground up cells, tissues, or other secretions. And some examples: blood agar, serum agars, meat extracts or infusions, milk, yeast extract, soybean digest, or peptone. Nutrient broth, blood agar, and McConkey agar all contain a rich mixture of nutrients for microbes that have complex nutritional needs. But again, each one of these has at least one component that I cannot say exactly how much is available to the microbe. Now, media can be used for different purposes. The general purpose medias grow as broad a spectrum of bacteria as possible. In other words, pretty much anything that would grow artificially will grow on a general purpose media. These are typically complex medias that contain a mixture of ingredients that support a wide variety of microbial life. Versus enriched medias. These contain complex organic substances that fastidious bacteria require for growth. What exactly is a fastidious bacteria? Well, it's fussy. It likes certain things in its media or it's not going to grow. And some examples would be something like blood agar right here, or a chocolate agar. Yes, there is a chocolate agar. They like chocolate too. And these contain certain growth factors and usually specific vitamins or amino acids or something else like the blood that's in blood agar. Now, what's the difference between selective and differential medias? Well, these are designed for special microbial groups. They have extensive application for isolation and identification. Both will permit preliminary identification of a genus or even a species of bacteria. When I see an emerald green colony growing on a MAC plate, I know that it's going to be E. coli. Selective medias contain one or more agents that inhibit the growth of certain microbes. In our laboratory, one of the main things that we use selective media for is to select against either a gram-positive or a gram-negative bacteria. So it will encourage a select microbe to grow while completely disallowing the growth of a different type of microbe. It's important in primary isolation of certain types of microorganisms that are in mixed samples. Differential media allows multiple types of microbes to grow, but displays visible differences between the colonies and or the media itself. Differences in colony size or color, changes in the media from the original color to now a different color, the formation of gas bubbles, or even precipitates within the media. Variations may be due to metabolism of certain ingredients that cause color changes. So, for example, here I have a mixed sample of, and if I were to plate that on a general purpose media, everybody grows. So I've got a whole bunch of different ones here. But on a select media, only this particular organism grows and everybody else will not be allowed to grow. Same scenario, here I have my general purpose mixture on a general purpose growth plate, and I've got different organisms and they all look very much alike. But if I were to put them on a differential media, I can see that actually I have three different species that appear different from each other on that differential media. So there is a difference that is shown between the different organisms. And this is just a short list of some of the selective median agents and their functions. We will use certainly the McConkey auger. We call those MAC plates. I don't know if we're going to use mannitol salt auger or not, but there'll be different ones that you'll see within the laboratory. This is just a couple of examples of differential media. This media is actually known as the Klegler iron media. This is a tubed solid media. This is the original color before anything is inoculated. And you can see that once things have grown, that we can sometimes have color changes. We can have yellows or we can have black. Differential medias, you'll notice over here that we've actually put on some different organisms and they all appear different from each other. So E. coli looks different than Klebsiella, looks different than Proteus or Enterococcus. Blood agar is a differential media, but it's also an enrichment media for the fastidious bacteria that require blood. Hemolysins are enzymes that lyse red blood cells to release iron-rich hemoglobin. Many organisms need iron in their metabolism, and so they will get it actually from the blood by releasing these enzymes. If we see beta hemolysis on a blood agar plate, this shows complete lysis of the red blood cells in the region where the cells are growing. Alpha hemolysis will see incomplete lysis of the red blood cells in the area where the bacteria is growing. And if we have gamma hemolysis, there is no hemolysis surrounding the growth of the microbes. So here's what it looks like. Here's the beta hemolysis, and you can see the original color of the media was this red color due to the blood. But you'll notice now that we have this yellow halo where the bacteria are growing. That is because all of the red blood cells in this area have been broken down by these hemolysins and completely hemolyzed. The alpha hemolysis, you'll notice that there's a small kind of shadow around the alpha symbol here, and this is due to partial hemolysis of the red cells. But down here at the gamma, we have absolutely no hemolysis at all. Now, some medias can be both selective and differential at the same time. And some of these are the McConkey agar, the mannitol salt agar, and this is what we're talking about. So, I actually can see that there are differences on here between these three different bacteria, but we also can see that there, for example, are no certain other ones growing. So for example, this might be all three of these may be gram negatives, but in the original sample there was gram positive, but it is not growing on this. So they're both selective and differential at the same time. So here's a really good example. This is a McConkey agar. And so I can tell you right now, this is E. coli that was inoculated. Again, the only time I'll see that emerald green colony is with an E. coli on a McConkey plate. E. coli is a gram negative. This is also a gram negative. This is a gram negative, but we inoculated a gram positive here. And you can see there's absolutely no growth. So this is selective because it's selected against the gram negative, or sorry, gram positive. It didn't grow. It only allowed the gram negative bacteria to grow, but each different type of gram negative looks different from each other. Miscellaneous media include the reducing medias, and these are going to be ones that will actually extract oxygen from the media so that we can grow anaerobic bacteria. Carbohydrate fermentation medias, where we put in specific sugars that can be fermented by some bacteria, and there's a pH indicator in the media that will show a fermentation. So for example, again, here's our uninoculated tube. We also have a small inverted tube called a Durham tube in this liquid media. We've inoculated both of these tubes with different bacteria. This one shows no growth of the bacteria or no usage of the sugar that was put in there because it remained the same color as the original uninoculated tube. This one, however, changed the pH, and we had a color change within the media. This shows that this particular bacteria used whatever sugar we put in here. And by the way, the Durham tube actually shows that this also produced a gas of some sort, and the little gas bubble gets trapped inside that inverted Durham tube. Transport medias, again, maintain and preserve specimens for transport. Assay medias can be used to test the effectiveness of antimicrobial drugs, and they're oftentimes used to assess the effects of disinfectants, antiseptics, cosmetics, preservatives, and other things on the growth of microorganisms. And then enumeration medias are used in industry and environmental microbiology to count the number of organisms in different things such as milk, water, food, soil, etc. Now, isolation, one of our big I's. A colony is a discrete mound of cells formed on a solid nutrient surface, usually a petri dish. And each colony was formed by a single organism, no other, uh, or a colony forming unit. So sometimes a single colony will be a single cell that begin to reproduce and grow. Or sometimes it will be a group of cells that are all identical to each other that form this colony. So we sometimes refer to each colony being made by a colony forming unit or CFU. So each one of these colonies on this plate either came from a single organism that began to grow and reproduce, or a small group of the same cells that produced each one of these colonies. Requirements of isolation. We first of all need a small number of cells that must be inoculated into a relatively large volume or expansive area of the media selected to encourage growth. It needs to usually be a relatively firm surface, such again as an agar in a petri dish, and inoculating tools such as inoculating loops or spreaders will be utilized. So here we have a mixture of samples. Okay? Each one of these cells could grow its own colony. How are we going to separate these? Well, we can simply separate the cells by spreading or diluting them into an agar media, and what we'll see each colony grew from a single cell or again a group of these cells so that now I could take each one of these colonies off of this particular plate, put it into other growth media, and have a what we call pure culture of that organism. The streak plate method is the most common method that's used. A small droplet of culture or sample is spread across the surface of a media using an inoculating loop. The pattern used to inoculate gradually thins out the sample and separates the cells in order to encourage the growth of discrete colonies on the surface media. So here's how we do a streak plate. First of all, you'll take your inoculating loop and you'll make a single pass. So on this plate, we just simply made a pass back and forth along the surface of this media. Then we sterilize the loop. Then what we'll do is we pull that loop through the edge of the first streak line that we did, and we kind of space it out a little bit more. And that's what we did here in our second streak line. Then we sterilize the loop, and we'll drag it through the second streak line and again kind of spread it out a little bit more. And that's what we did here. And then sometimes, depending on the size of your Petri dish, we can even do it a fourth time, sterilizing the loop, dragging it through the third streak line, and now bringing it out to a fourth streak line. What this does is, in effect, dilute the number of cells each time we make a new set of streak lines. And so what we'll find is out here in the third or fourth streak line, we begin to see single colonies. Now I could take a sterilized loop and pick that up and put it into another media. The loop dilution or pour plate method is similar in that it dilutes the sample, but what we do here is we actually dilute serially into cool but still liquid agar tubes. The inoculated tubes are then poured into sterile Petri dishes and allowed to harden. The diluted cells then will have enough space to grow into separate colonies. One disadvantage to the pour plate method is that some colonies may, however, grow deep within the media rather than just on the surface like we would see with the streak plate method. So we simply do this. We will dilute into each tube. So we may take a tenth of this and put it in with 9/10 of a fresh media, and then we'll take 10 of that and put it in with 9 of this. And so each time we're simply diluting, and we simply pour then that liquid media that's not quite set up yet, and we will get this distribution of the colonies. The spread plate method is somewhat similar to the other two in the fact that we will put a small volume of sample pipetting it onto the surface of an agar plate, and then using a sterile metal or glass rod, we simply spread the diluted sample around on the surface of the media. This will dilute out the cells. And so we put a small amount on, we use this hockey stick, either metal or glass sterilized rod, and just simply spread it all over the surface of the plate. A pure culture then is anything that contains a media that contains only a single known species or type of microorganism. This is used most frequently for laboratory studies. So when you get your unknown, the first thing that you're going to want to do is streak for isolation so that you can get a pure culture of your organism. If you have 2 or 3 different organisms, you're not going to be able to identify the single organism we want you to. Anexic is simply a word that says something is free of other living things except for the one that's being studied. A subculture is a second level culture from a well-isolated colony. Many times what we do to make your stock cultures that you're going to utilize in the laboratory is we will streak plates, streak for isolation. We will get a single colony and we'll put it into a fresh broth media. That's a subculture from the streak isolation plate. Mixed cultures hold two or more identified, easily distinct, differentiated species of microorganisms. So we may have two different things within our media that we're looking at. However, what we want to stay away from is a contaminated culture. And this is a culture that was once pure or mixed but now contains contaminants or unwanted microbes of uncertain identity. We don't want friends in our cultures. So here's a nice little streak plate, and what we can see that we have two different types of organisms, one that produce red colonies and yellow colonies. So there's the reds, and there's our yellows. But here we have a friend that is growing on our plate. This is the bacteria that we want, but this guy is something else. And in fact, it happens to be a fungus that also is growing on this plate. This is an unwanted contaminant within our agar. Now, inspection and identification. Microbial profiles are determined through combining several things. One, we're going to look at the phenotypic testing. What does the organism look like? What does the colony look like? We can do genotypic testing. What is the actual genes of these organisms? We can do immunological testing, macroscopic analysis. What does the microbe look like? And microscopic analysis. How does it test in the gram stain? And then we can use biochemical tests that can determine fundamental chemical characteristics such as the requirements of nutrients that this organism needs. What products may be given off during growth? Does it produce a gas? Does it produce something that causes a color change within the media? Are there presence of certain enzymes that the organisms will produce? And what is the mechanism for deriving their energy? How do they make their ATP? Some other analytic and diagnostic tools can be that genotypic testing. We actually can detect microbes based upon their DNA signature. And with the advent of polymerase chain reaction, PCR, this has become much, much easier since in the last few decades. Immunological testing tests the isolate against known antibodies, and we use a lot of these in diagnostic situations, and inoculation of a suitable laboratory animal. Now, we are working with biohazardous material. While the microbes we work with in the laboratory are not exactly extremely pathogenic, they can be. So here's a rule of thumb for the lab. Do not lick the E. coli plate. Okay? Just don't do it. Cultures and specimens collected from patients or environments that are grown become biohazardous. Steam sterilizing or autoclaving or incineration are used to destroy microbes. This will get rid of everything that could potentially cause disease. So here's a concept check. Can you define two definitions of the term culture in regards to microbiology? What are the three physical states of the media? What's the differences between selective, differential, or enriched or general purpose medias? And list and describe two types of media that are both selective and differential. Now, in microbiology, size matters. And dimensions of macroscopic organisms are given in terms of meters and centimeters. In science, we operate with the metric system. But because what we're looking at here in microbiology are microscopic, we have much smaller units of measure. So dimensions of microscopic organisms are measured in millimeters to micrometers or micrometers, or microns is another term, to nanometers. With the advent of the technology in microscopy, however, we can now even go down one further, and that is into the picometer range. And so here's a nice little picture. So while we're looking at these values right here, so we're looking oftentimes at centimeters. Okay, and if you look at our ruler here, each one of the longer lines is a centimeter. You'll notice, however, that there are 10 little lines between each of the longer lines. Each one of these is a millimeter. So there are 10 millimeters in each centimeter. Micrometers, or microns, if you were to take the space between two of those little millimeter lines, and my pen just isn't small enough to even show those, but if you were to put 1,000 hash marks between 2 millimeter lines, each one of those would represent a micrometer. Okay. So there are 1,000 micrometers in each millimeter. And then if you were to go in between each one of those micrometer lines and put 1,000 hash marks, each one of those would be a nanometer. So there are 1,000 nanometers inside each micrometer. And if we do the math, that means that there are 1,000,000 nanometers in each millimeter. So put 1,000,000 marks between each of those two little millimeter lines. There you have nanometers. And if we go down one more into picometer range, there are 1,000 picometers in each nanometer. So that would mean that there are 1 billion picometers in each millimeter. We won't get down into the picometer range, but because, again, of our technology in microscopy, we now actually are getting down into the picometer and femtometer ranges. So let's look at some comparison here. So here's the red blood cell. It's at about 7.5 micrometers in diameter. Our tick, an adult Ixodes tick right here, is at a little over 1 millimeter in diameter. So you can see there's quite the size variation from the red blood cell to even the tick. But let's look at some of our microbes. So here, E. coli is at about 1 micrometer in diameter. It's about 3 micrometers long. One of the viruses, our T bacteriophages, is just a little over 100 nanometers in length. And then with, again, our microscopy, here is a piece of DNA, one of our chromosomes, little over 1 nanometer long. The coronavirus is actually 30 nanometers, or around 20 to 30 nanometers in size, so the coronavirus fits right there. So protozoa and algae, about 3 to 4 millimeters. Yeasts are about 3 to 4 micrometers in diameter. Some of the smallest bacteria are 200 nanometers. That would be 0.2 micrometers in diameter. And viruses tend to range somewhere between 20 to about 800 nanometers. In fact, the coronavirus is actually one of the smallest viruses that we see. Our eyes can actually distinguish between very small things up to about 1 millimeter apart from each other. It's been estimated we can go down to even about 0.2 millimeters, but that's about the size or the smallest size that we can distinguish two distinguishable points. So that's why we need to go to the microscope. In the light microscope, we can actually get down to about 1 micron in diameter. So how big are your bacteria going to be on your microscope slide? They're itty bitty little guys. So you have to get up on very high magnification, and we'll talk about that. With our electron microscopes, however, we can actually get down to the 1 nanometer range. And with some very specialized microscopes, we can even get down to 0.1 nanometer. That is 1 angstrom. And what we can then actually visualize with some of these very specialized microscope is even the atom itself. So what exactly is magnification? Well, this results from a complex interaction between visible light waves and the curvature of a lens. And so here we see a couple of different magnifying lenses. Here's just our good old magnifying lens itself. And then you'll notice that this glass ball, though, actually has magnification abilities as well. We have to have a curvature of a lens. So let's look at the effects of magnification. So what we're going to be looking at is this little guy right here, and this is magnified 7 times. So we can see that there's some other bugs next to this little guy. But at 50 times magnification, we can get up kind of close and personal with his face. So we're looking at this area right here on the lower magnification. And you'll notice that as we go from 7 to 50, that we exclude other things that are on the periphery of the lower magnification. Now we're getting up even closer. This is actually his eye right there that we're looking at 220 times magnification. And then finally, at 950 times magnification, we're actually looking at the surface of the eye. One thing I want to point out then, as we increase magnification, our field of view gets smaller and smaller. Here we've got a very large field of view. We can see everything. Here we're narrowing it down. Here we're going even further down. And now we're actually looking at the individual components of this eye on this bug. Another characteristic of light waves that we have to talk about in magnification is refraction. And refraction is simply defined as bending or the change in the angle of the light ray as it passes through media such as a lens. But we can see this principle demonstrated here in the glass of water. So this is a spoon and it looks like it's bent. Is it? Well, it's not. What we're seeing is light refracted through the different medias. We can actually see that there's a slight bend here as we go from the air to the glass. And then we see a really big bend when we actually go from the glass into the water. And so this is actually refraction. The other thing I want you to notice is that blue is on this side when we're looking at it through the air and even the glass. Yellow's on this side. But what happens once we get down to the water itself? These are inverted, and refraction does that. If you go back to your eye, the lens of the eye actually inverts the image that we're seeing to be upside down on the retina of the eye. Is that okay? Sure, because our brain interprets it as being right side up. The greater the difference between the two substances, the greater the refraction. So there's not a huge difference between the air and the glass. So we don't see a huge amount of refraction. But there is a great difference between the air and the glass here and the water down here. And that's why we see this inversion of the image as well. So formation of an image occurs when the object is placed at a certain distance from the spherical lens and illuminated with light or another illumination source. And it depends on the curvature of the lens. in how the image is enlarged to a certain degree. And this is what's called the power of a magnification, or its X, or times. So, as in with the bug, we said that it was magnified 7x. It's magnified 7 times its original size. Now again, Leeuwenhoek with his first microscopes, and even going back clear to Robert Hooke, was very simple magnifying lens with very few working parts. But with the advent of Leeuwenhoek's microscope with the adjustable stage, we then came into what we now have as the compound microscopes. And now we have usually a second, third, or even fourth magnifying lens that's part of our system. There's lamps in the base to illuminate the specimen, or again, another illumination source that we can use besides visible light, and a condenser that's used to focus the light or other illumination source onto the specimen. So here's the good old microscope that you'll be using in lab. It's very similar to the one you'll have. And so you should refresh yourself with the different parts of the microscope. Depending on what microscope you have, the control, the focus knobs will be usually located somewhere else. The on and off switches will be somewhere else. May or may not have a light intensity control, but they'll have condensers. They'll usually have 4 to 5 objectives on the revolving nose piece, and we can have 1 or 2 ocular pieces. We have binocular scopes, so you do need to be able to use both eyepieces or ocular pieces. Some microscopes actually have a third ocular that can be hooked up to a camera or to another type of media that can then visualize what's being seen on the microscope. So 3 properties of an effective microscope are going to be magnification, resolution, which how clear is the image, and contrast. And this is just the pathway of light. So here's our illumination source, in this case visible light. It will go through the condenser lenses, and you'll see the light gets refracted. It will then hit a focal point on the specimen on the slide on the stage. That then spreads out and will go through the objective lenses, will be refracted again through a prism, and finally refracted through the ocular lenses. So your image is going to be upside down and backwards by the time it gets to your eyes from what it is on the slide. The ocular lenses have a fixed focal point of 10x. That means if you just look through the ocular and you got it to the correct focal point, the image would be enlarged by 10 times. Known also as the eyepiece, but we would prefer that you actually use ocular. This forms the virtual image, what your eye will actually see, and the eye will then convert it into retinal and visual images. The objective lenses are closest to the specimen, and on your microscopes we have a revolving nose piece with 4 of these objective lenses. They form the real image that then will be refracted through the prism and the ocular lenses. The pathway of light and the 2 stages of magnification in a compound microscope, then, we actually will see basically 3 focal points. One will be on the specimen through the condenser, so that's a focal point. From that point, though, the light waves will spread out, and we get a second focal point somewhere through the objective and the prism that spreads out again. Then the oculars then will focus that image on the back of your eye so that you should see a clear image. The magnification power of the microscope is calculated then by multiplying the power of the ocular lenses, which is always set at 10x, by the power of the objective lenses. So on our microscopes, we have the scanning objective, which the scanning objective lenses by themselves only offer a 4x magnification. So if we multiply 4x times the objective magnification, that's 40x times total magnification. The second highest power is the 10x or low power objective. It is a 100 times total magnification. The next one is the high power dry objective, and its total magnification is 400x. And then we have the oil immersion objective, which by itself is 100 times magnification, so that gives us a total of 1,000 times magnification. Resolution and resolving power is the capacity of an optical system to distinguish or separate between two closely adjacent objects or points from each other. I said that our eye by itself, depending on how good your eye is, can actually distinguish down to about somewhere between 0.2 to 1 millimeter of separation. So we can get about to that area. But microorganisms cannot be seen by the naked eye. So what we're going to have to do is allow our microscope to do that work for us. So what's in this image? Well, you'll notice there's a lot of little boxes that make up this image. All images are made up of little boxes called pixels. Now, I can actually distinguish between each of the adjacent pixels in this picture, and therefore it is very fuzzy or blocky. However, if I reduce the size of the pixels to where I can no longer distinguish between those adjacent points, I get a much clearer or resolved image. So resolution is determined by a combination of the characteristics of the objective lenses and the wavelength of light being used to illuminate the sample. Optical microscopes use visible light or the visible light spectrum. And this actually shows the visible light spectrum in the electromagnetic spectrum across the board. The higher the frequency of wave, in other words, the closer the waves are together, the more energy that is produced by these waves. Visible light ranges from about 400 nanometers on the violet or blue end to about 750 nanometers on the red end. If we look at white light, all of these wavelengths are in white light. White light is the combination of all the colors of the rainbow. In your microscope, in the condenser, there is a blue filter. What that blue filter does is this. It basically allows the blue or higher frequency waves to go through while excluding a lot of the lower frequency waves or down here in the red spectrum. The higher the energy, the greater the resolution power. So what do we want? We want more of the blue end of the spectrum because they're higher frequency waves than the red end. That's why when you just look in your microscope and nothing's on it and you just see the light, it kind of has a little bluish tint to it. That's because we're allowing more of the blue wavelengths to come through than the red. So resolution power, low resolution, you get this kind of fuzzy image because again, we're having to distinguish between larger blocks. Whereas now, if you get a smaller block, then we get a much higher resolution and better focusing or better imaging. Now, let me talk about the oil immersion lens. A drop of oil must be placed on the slide when using the oil immersion or 100x objective lens, and I'll explain why here in just a second. The oil that we use is not just any old oil. It has the same optical qualities as glass. The oil then prevents the scattering of light waves and increases the numerical aperture and resolution. The oil immersion can resolve objects that are about 0.2 microns apart. So this is what we see. What we'll see is on the side where we don't have any oil, as the light comes away from our image, some of the light waves get scattered and don't enter the very small aperture of that oil immersion objective. When we put the oil on, however, it keeps some of those light waves from being refracted outward and actually then will be captured inside the objective lenses. And so this will allow, again, a better resolution of the image. Sometime just take a look without oil on your slide and you'll notice it's really, really fuzzy. Then when you put oil on, it becomes very much more clear. Now, refractive index simply means a measurement of the degree of contrast between a magnifying image and its surroundings. So we need to talk a little bit about contrast. Refractive index refers to the degree of the bending that light undergoes as it passes from one medium to another. And the higher the refractive index, the greater the contrast will be. So let's look at some of the types of light microscopes that we can use. The bright field light microscope is what we will be using in the laboratory. This means that we are using all of the visible light spectrum, again, more of the blues than the reds, but we see basically white light. And so specimens will be dark against a bright background. Dark field light microscopes are the same microscope, except what we do is we take an opaque disc and put it in the center of the condenser. What this does is blocks all the center rays from coming through, and we only get the edge rays coming through. What this will create is a dark background, and our objects will be lighter against that dark background. Phase contrast microscopes, most of you will never see one of these, but what we use is we try to get a lot of those wavelengths of the different light spectrum that's coming through hitting at the same time. They're peaking and troughing at the same time. And what this will do is actually give you good contrast, especially on the surface of your subject. And then differential interference phase contrast. This is where we're actually using two light sources, one that is perpendicular and one that is horizontal to the specimen. And I'll show you some examples of these here in a moment. Fluorescent microscopes actually use the ultraviolet spectrum just outside of visible light. And so on the specimen, we're going to have to use something that will fluoresce. So we'll use fluorescent dyes. And then we can pick them up in the visible light spectrum. And then confocal microscopes actually uses a laser beam as an illumination source. Our maximum practical magnification is around 2,000 to 2,500 times magnification. So you can see that the outside limits of our light microscope is actually very small. So what did we do? We came up with a new illumination source, and that's electrons. The electron microscopes can magnify very high in their magnification. Our transmission electron microscopes can go up to 100 million times magnification and have a resolution of a half a nanometer. This is really, really good for details of very minute organisms. The scanning electron microscope goes again to about 100 million times magnification. And it's really nice, and I like the name because it gives you nice surface area of these organisms. But you'll notice it can only go up to a resolution of about 10 nanometers versus the transmission electron microscope at a half. And so if I want to look at surface areas, I can use that scanning electron microscope. If I want to see more internal structures, I'll probably go with the transmission electron microscope. And then I said we had some very specialized microscopes that could even go up to the size of an atom, and these include the atomic force microscope and the scanning tunneling microscope. In both of these, they use either a diamond or metal tip, or in the scanning tunneling, we use a tungsten tip. And what we'll see then is, yes, we get nice resolution and nice magnification, even down again to the size of an atom. But here's the disadvantage of these two. We destroy the specimen in the process of the magnification. And again, probably none of you will ever see an atomic force or scanning tunneling microscope. So I'll show you some examples of these. This one is actually a dark field microscope. And what we'll see is, again, we have light organisms, this is a big cell and these are bacteria, against a dark background. This is actually a scanning electron microscope of the scabies mite. And I love this picture. This is actually skin cells, and this is the surface of the mite. And one of the classic signs of scabies is that you itch and itch and itch. And the reason for that is you'll notice these little kind of looks like hairs. They're not, they're spikes. And these guys are crawling around in your skin. This one's actually upside down here. And so as they're crawling around in your skin, they certainly cause a lot of irritation. This one actually is a phase contrast microscope, and this was very interesting just simply because of the item that we're looking at, and that actually is a snowflake. And then this is a transmission electron micrograph of chromosomes. Okay, so how do we prepare the specimens? Well, the method of specimen preparation will depend on several things. One, the condition of the specimen. Do we want to look at it in the living state or the preserved state? Now, let me just say this one thing. To look at a living state organism, you cannot stain it. Stains will kill a living organism. So if you want to look at a living organism, stains are not going to be something that we can choose. What are the aims of observing this organism? So is it just observation? Do we want to identify it? Do we want to identify how it's moving, if it moves? And what is the type of microscopy available for me to utilize? Wet mounts or hanging drop mounts, this is where cells will be suspended in a suitable fluid, usually water, broth, or a saline of some sort, and the fluid maintains viability of the organism and provides a media for movement if the organism moves. So it provides a true assessment of the size, shape, arrangement, color if there is any, and motility if there is any of the cells. Wet mounts will consist of a drop or two of the culture placed on the slide, overlaid with a cover slip. The advantage of wet mounts is they're quick and easy to prepare. The disadvantage is it can damage larger cells. They're susceptible to drying and can contaminate the handler's fingers because these will be living organisms. So how do you prepare one? They're prepared with a concave or depression slide, so it's a specialized slide that has a concavity ground into it, a Vaseline adhesive or sealant, and a cover slip. The reason for the Vaseline is to decrease drying out. So this will overcome some of these disadvantages of the wet mount. And you'll see the hanging drop and be able to focus up and down then in that drop. Fixed stains or smears. The smear technique is we're going to spread a very thin film made either from the liquid suspension of a cell on a slide. We're going to allow the cell to air dry or heat dry, and then we have to affix the cells to the slide. We primarily will use heat fixing, which means that we heat the slide gently after it's been air dried, or you can use chemical fixatives. And so here we're applying a thin layer. And let me state that again. You want a thin layer. The thicker layer is harder to see through, so you want very thin. And depending on whether you're using a liquid or a solid, which we can use solids, but you want to get that solid spread out very thinly. Important functions of heat fixing is that one, it will kill the cells. Two, it secures the specimen to the slide so that when you do stain it, the slides do not come, or sorry, the specimens don't come off the slide. It preserves cellular components in a natural state with minimum distortion. And again, alcohol or formalin or other chemical fixators can also be used as well. And this is a stain I actually use in clinic, and it's a three-step stain. This clear right here is actually an alcohol acetone mixture, which is our chemical fixatant. So we put the slide in it first. That kills and fixes the cells to the slide, and then we can then stain it in the other stains. Staining. Why do we stain? Well, one, it provides a contrast. Two, it makes inconspicuous features stand out. Dyes impart colors to cells by becoming affixed to them through a chemical reaction. So dyes that we use in staining are either basic or catatonic stains. They will have a positive charge and therefore will be attracted to acidic negatively charged components of bacterial cell walls or other things in cells. And the acidic or anionic stains, which have a negative charge, they're repelled by the acidic negatively charged components and then will actually be attracted to anything that's positively charged in the cell. A positive stain will be attracted to negatively charged cell walls, and they stick to the cell and give it color. Whereas negative stains, negatively charged dyes are repelled and therefore produces a black background around the cells. So here's the difference. A positive stain, the cells actually take up the color and are contrasted then against the background. So the background does not stain. Whereas a negative stain actually will stain the background of the slide while not staining the actual cells. So what we see is basically voids in the blackness of the negative stain. So here's a positive stain. So our microorganisms take up the color while the background stays light. And here's a negative stain. Now, we have stained the organisms. That's the organisms right there. But what we did is we stained the slide itself. And so this pinkish color is actually the negative stain. And why we did this is we used a positive stain on the cells and a negative stain on the background so that we could see that halo that's around each one of those microbial cells. And that is actually a structure known as the capsule. And this is a capsular stain. Simple stains have one dye, either a basic or an acidic stain. It's a very uncomplicated process. You either put the stain on the slide and you'll usually wash it off to wash any off the background, or you'll just dip the slide in the stain. Differential stains or complex stains have two different colored stains, usually one basic and one acidic, that clearly contrast cell types or cell parts. And so complex staining techniques have a primary stain, which is the first stain applied, and a secondary or counter stain that will be used. The Gram stain is a differential stain, and it is going to be your guiding light in microbiology. All bacteria are either gram positive or gram negative. And this is one of our big separations that we use for identification. Acid-fast stain, this is a differential stain that looks at a particular group of microorganisms called the acid-fast bacteria. Endospore stains will highlight endospores in those bacteria that can actually produce endospores. Capsular stains, just like I showed you a second ago, will highlight the capsule that some microorganisms can produce. The Gram stain was actually developed by Dr. Gram, and it was developed over a century ago, and it really remains the universal diagnostic staining technique for bacteria. It is very important that I know, is my bacteria in my patient a gram positive or a gram negative, because that's going to affect how I treat the organism in my patient. It permits a very ready differentiation of the major categories based on the color reaction of the cells. And gram-positive bacteria stain purple, while gram-negative bacteria stain a reddish pink. This is the way I always remembered it. Gram-positives, purple. The two P's go together. Gram-negative, red. Red has that negative connotation, and that's the way I remembered it. Now, the first thing that we're going to do is we're going to place our smear on our slide, and we're going to heat fix it. This basically then effectively fixes the cells to the slide. Our primary stain in the gram staining process is crystal violet. We'll flood the slide with crystal violet for 1 to 2 minutes, depending upon what procedure you use. And everybody stains purple. Whether you're gram positive or gram negative, everybody stains purple. So if you were to look at the slide after the application of the crystal violet dye, this would be a simple stain at this point, and every cell is going to stain purple. Then we're going to wash off the excess crystal violet with deionized water, and we're going to add iodine. Iodine is a mordant in this case. Iodine, even though it does have a color to itself, does not add color to the cells. That's why it's called a mordant. Think of the mordant as a highlighter. It actually will make the cells all look more purple at the moment. And the reason for this is the iodine will enter each of the cells and combine with the crystal violet. We then will wash off the iodine after 1 to 2 minutes. And then the critical step of gram staining is this right here, and this is known as the decolorizing step. We are going to now wash these slides with an acid alcohol solution. And this says for 20 seconds. That's going to be too long, folks. What you want to do is simply drip this on and then immediately wash it off with deionized water. If you were to look at the slide at this point, some of the cells will have, quote, disappeared. They will become colorless. The reason for this is the cell wall. Gram-negative cells will lose the crystal violet iodine through their cell walls, while the gram-positives retain the crystal violet. Then we're going to apply the secondary or counter stain known as safranin, and everybody will take up the safranin. But since the gram-positives are already so purple, if nothing else, these gram-positives now look even more purple at this point. Okay? We will not see the red of the safranin stain. But since the gram-negatives became colorless in the decolorizing step, they take up the safranin and appear reddish in color. And this is a gram-stained slide. So these guys out here, okay, what is their gram reaction? And then looking at these fellows, what is their gram reaction? So we have two different organisms, and you can see they're even different shape. These are kind of a sphere shape, and these are elongated. And so what are the sphere shape gram reaction, and what are the rod-shaped gram reaction? Now the acid-fast stain differentiates acid-fast bacteria from non-acid-fast bacteria. Acid-fast bacteria will stain pink in the acid-fast stain, and the non-acid-fast bacteria will stain bluish in color. These detect agents of two genuses of bacteria, the Mycobacterium and the Nocardia genus. In Mycobacterium, we have the causative agents for tuberculosis and leprosy. So, our primary stain in the acid-fast stain is carbofuchsin. It is an acidic red stain. We then are going to decolorize with an acid-alcohol mixture, just like we did in gram stain. And our counter stain then will be methylene blue, which is bluish in color. The acid-fast organism takes up the carbofuchsin and will retain the carbofuchsin throughout the rest of the steps of the acid-fast staining technique. But non-acid-fast organisms will be decolorized and lose the carbofuchsin in the decolorizing stage and then take up the methylene blue. So this is what we get. So if this was a sputum sample from my patient that's got coughing and I'm suspecting Mycobacterium tuberculosis and I ask for an acid-fast stain and I see these acid-fast pink stained reddish elongated bacteria in my patient's sputum, I pretty much have my diagnosis of tuberculosis. All the other cells here, and these are some other bacteria up here and even here, they lose that carbofuchsin and take up the methylene blue. Endospore stains are used to distinguish endospores, which is a survival mechanism that some genuses of bacteria can produce, from the vegetative or living cells. The two major pathogenic classes of endospore formers are the genera Bacillus and Clostridium. What we do with this is our primary stain is malachite green. We will actually not heat fix our smear at this time. We will apply the malachite green and then heat fix it. What the heat does is fix it and also acts as a mordant. At this point, everybody stains green. The smaller round things are the endospores, and the larger one are the living or vegetative cells. We then decolorize with just deionized water. If the living cells are still present, they will decolorize and lose the malachite green. And so all that will be left in the staining process will be the green endospores. We then use safranin as our counter stain, and now we can actually see that anything that was living will take up the safranin, and you'll notice that in this case we actually have endospores still forming within the vegetative cells, but we also have just some free ones that are floating around. And so what we get is this. So here we have endospores that are formed within the vegetative cells. Here's actually probably one that's already lost its living cell. And these endospores actually can then withstand a whole bunch of unfriendly environmental conditions like heat or cold or dryness until the environment becomes favorable again, and then each endospore will literally hatch out and grow a new cell. Endospores are not reproductive spores. The capsular stain. This capsule is an unstructured protective layer, usually of proteins and sugars, that surround the cells of some bacteria and some of the fungi. These are visualized through negative stains with India ink or specialized positive using negative stains as well. And they're very useful in identifying such pathogens as a particular fungus known as Cryptococcus. And this is actually the Cryptococcus organism. It is a yeast. It is a round body. And in this case, what we've used is an India ink stain, the negative stain only. And you can see that the Cryptococcus has a huge capsule. The capsule does not take up neither a negative stain, nor if we used a positive stain to stain the organism itself, the capsule takes up neither one. So we see it as basically a halo or a ghost around the cells. Flagella. These are tiny cylindrical filaments of protein used by bacteria for locomotion, and we'll talk about them a little bit later on. The flagellar stain, because these flagella are so thin, they have to really be highlighted in order to be seen. And so what we're going to do is we're going to use a mordant, and the mordant in most cases is going to be iodine again. And this coating or highlighting then is deposited on the outside of the filament and enlarges it enough so that we can actually see it. And so here's our living organism, the elongated rod here. And then you can see these kind of tiny little hair-like structures coming off, and these are the flagella. And so this is a flagellar stain. Okay, so the last concept check here. What is the total magnification of the specimen viewed with the 40x objective? What is the importance of resolving power? Describe the difference between simple and differential stains. And what is the purpose of the gram stain? Thanks. Have a great day.